All right. Can everyone hear me okay? Those online.
Yes.
Okay, thank you. Welcome everyone to this class. Thank you for being here. Thank you for being online. There are more people online than being here, it looks like. And we have one more people joining us. Okay, I guess two more. Okay, welcome.
Please show up on time, please.
So yeah, thanks for being here bright and early. So this class, actually, uh, it's called Spatial Transcriptomics. Life is because life is short and full of distractions. If you just look here and there, but it's something to commit 3 days to this and focus on what's called spatial transcriptomics. And it's funny that my laptop thinks this is a typo, like transcriptomics. It doesn't recognize it. What does it mean? It thinks collaboratory is a typo too. So anyway, so we will learn. But I hope that by the end of 3 days, you think it's a good distraction. And we have 3 goals to for this workshop. First is just to learn something about this term, spatial transcriptomics. It's not a typo, it's something new, definitely. And we will practice coding in the context of spatial transcriptomics data, and we will chat with each other, and hopefully you meet someone who shares similar goals with you, so you can help each other in the future. So that's pretty much it. 3 goals for the 3-day workshop. So let's start from the last one, chat. Let me introduce myself. I started teaching this class about three years ago when I was a postdoc. I'm now still a postdoc, but now live in a different city, so I have to fly in yesterday. And back then, they put me in on Thanksgiving week. You would think, who would have shown up on a Thanksgiving week for a 3-day workshop like this? But it turned out someone showed up and it worked well. And if you have seen the YouTube video for this class, it has been one of the most popular workshops for collaboratory. But anyway, so now we moved it to the Halloween week. Hopefully we can attract more normal looking people. I studied a lot of things. I studied physics in the beginning, then I studied genomics, I studied data analysis by being a bioinformatician. Now I study neurodevelopment. And it turned out spatial transcriptomics is really useful, a powerful tool to study neurodevelopment as well. So that's so much about me. So for the first 5 minutes, I would like you to talk to your neighbor. Find one of your neighbor and just say who you are, what do you study, and what brings you to this class. What do you want to accomplish? So just find a pair. Yeah. And for those of you online, feel free to type out who you are and what do you study. And by the end of 5 minutes, we will take a poll.
So this is geared straight. Yeah, well, right on the nose. Yeah, I've never done it before. Yeah, it's lost state. Yeah, need some orientation. Like everyone, yeah, and I'm like. So I'm talking to yeah. So I'm making you sort of large scale bioscience studies. So you're ready to leave. I'm just doing revision. There's a lot of methods, but I'm always applying methods that are available, so I just like that. No, like and you never know how it is. However, conflicts that but I think it's also just good orientation, like even if you don't use it, if you have papers that then use it, I think it's pretty. Yeah, because I'm sure that when we try to find something and we try to study at some point, it would certainly. So do you work with human tissue? Do you work with just human data or? That's my work. Right?
Yeah.
Gotcha.
Yeah.
Yeah. Awesome.
If I forgot, let me know. I should. Yeah, I will. Yeah.
It's perfect. But it's kind of like yeah. We want to function validate that so that we have yeah. Okay, you remember the mom's data. Right. Yeah. Oh, okay, so you also look at that. A little bit, but I really am sure. But yeah, I can analyze more data. Yeah. No, that's I feel like I don't because they know that there are.
Yeah,
they did on that.
Exactly. This is what I'm hoping for that. Yeah.
There's just more and more.
I mean,
We're using your text platform. That's the best thing to know what you want. And stuff. And so I like.
Of course.
Because we see
where and double check where
the spatial aspect, especially
yeah.
It matters, like where you are. Matters. I can't. That's the reason I start doing like back sorted already sleep or something. But it's just like, but then I don't know how far away from the order I am. Right. Even if I collect like almost perfectly.
Yeah.
Without
That's also true with the cardiovascular disease. When you have a lesion, where about, you know, like, do you reset it or are you in the middle? Are you kind of at the end?
Is it only that?
Yeah.
You're like, why is this weird? Yeah. Yeah.
So
I'm very excited.
All right. Let's come back. We'll have more time to talk in the future. Back to the class. Thanks. It's very fun to talk to your neighbors and people who come to this class. Thanks those of you online who typed out a short introduction of yourself. So yeah, so basically that's it. That's our third goal accomplished. So we'll do a quick poll, including those of you online. If you can turn on your camera just for the next couple, like 5 minutes, that would be great. We'll do a short poll. So how many of you are undergrads? Okay, one undergraduate students. Okay, so here we have undergraduate student, one undergrad, 3 graduate students, right? Postdocs. Okay, 2 postdocs. That's 1, 3, 2, 6 people. Is that everyone? Did I miss anyone? Faculty. Oh, welcome. Normally we don't get that.
Yeah.
Great. Yeah. Okay. Yeah. For those of you online, if you're undergrad, please raise your hand or, you know, raise your digital hand. Graduate student. Okay, I saw a few hands. Postdocs. Okay, great, thank you. It's hard for me to count online, you can see, but it's okay. So you see we have undergrad, graduate students, postdoc, faculty. How about your major? How many of you study, say, something related to neuroscience in your work or in your, okay, 2, 3. How about cancer? 1. How about, I don't know, molecular biology in general? 1. medicine, maybe.
Pharmacy.
Yeah. Okay, you see, we got people from everywhere, from different level. That's the characteristics of this class. So that's the poll and chat. Hope we can learn from each other. And why do we do this counting? Because single cell genomics is all about counting. We develop different tools to help us count more comprehensively, more accurately. We use data analysis to try to analyze these counts better and faster, more efficient. But it's all about counting. Here we just count how many people are in different major. For single cell genomics, it's all about counting how many cells expresses this particular RNA, this gene, and how many copy of that gene is in a particular cell. The more accurately you can do it, the better you understand the underlying biology. And different technology will do it either more accurately or poorly. So like you guys online, I can't really tell how many because you didn't raise your hand at the same time. That would be a poorer technology to play with. So anyway, so no offense. That's one of the goal accomplished. So today and for tomorrow and Thursday, we will do coding. So we will leave this aside. Today, we'll just really try to focus on the first goal, which is to learn spatial transcriptomics. Okay, the basic concepts of it, and we will read a few papers about it. So let me just remove all of these. So we will, in particular, try to figure out 3 questions. What is spatial transcriptomics? Why do we need it, and how is it done? Okay. Okay. So this is an invitation to spatial transcriptomics. This term did not exist before 2015. So you can see it's a typo. My laptop still thinks it's a typo, but it really took off in the last 5 years or so, and it's growing rapidly, impacting all fields of biology. But it also might mean that everything you learned here will soon be obsolete. What's the point? If a field is growing, the knowledge you have is not stable. It's not like learning physics or chemistry or math, which was in most of the foundation were invented about over a century ago, and many people have organized it, so it makes no difference. Now you learn it or in 6 months, but in 6 months, I'm sure this field will be different for sure. So why do we still study it or be here? So being a scientist, our intuition often guides us to choose which subject to pursue. One of these are 2 quotes from 2 scientists. The first one is from a physicist, a theoretical one, which I was. He says, "Go for the masses. That's where the action is." He thinks physics has been developed for centuries and centuries. Newton came, Einstein came, all different people came to address different problems. But what, as a young scientist, would you choose? He advised that we should go for the masses. That's where the action is. That's published in the Nature called Four Golden Lessons in 2003 by Steven Weinberg. And the second one is from a biologist. You see all different kinds of science have similar thinking. He says what's known for certain is dull. I forgot who was the biologist who said that, but similar thing. If you know a subject really well already, it's harder for you to contribute. But if it's still ongoing, maybe you can do something with it. Learn what's the state of the art and try to jump in. So goal for this class is after it, you will not become an expert because arguably nobody is. We just try to figure out what's going on here. But you will have jumped into the deep water. I will give you some data sets to play with. We will read some papers together. And hopefully that brings you to the upfront, to the front line, and you either swim yourself or sink into the water. But it's okay if you sink into the water. It's not life-threatening. You could always either try it again or think for yourself, maybe you should try something else because there's plenty of exciting things to do. For day one, it's an overview. I will give some backgrounds, and there will be just about the basic concepts of spatial transcriptomics. And there will be 2 reading sessions. I have sent you emails containing 3 papers. We may have time to read the first 2 papers, definitely the first paper, more carefully together. And there will be a mini quiz after the paper reading, just for fun. It's not graded. If you're concerned about grading, don't worry about it. It's not going to be graded and usually the grading part for this class will be super easy. It's only for learning and your curiosity. Okay, and day 2 and 3 will be coding devoted to, where I will show you some data analysis techniques, show you some examples, and there will be hands-on practice in data analysis. I will give you time to do it. You will have a chance to share your results and ask questions. That's pretty much it. For those of you who need grading, there are 3 parts due next Friday. 1 part, attendance. Eloy will take care of it. There will be 2 assignments taken home. One is a quiz, the other is a homework. Together, they won't take you more than an hour to do, but if you happen to spend more than an hour, just let me know. Maybe there's something we could figure out together. I wanted to highlight that this class encourages you to ask any question at any time. There's no silly question. Don't worry about interrupting me. And in fact, the previous recording of this class has been online for more than 2 years, and many people have watched it. If you are interested, you can watch it too. It will be similar. But the only difference, perhaps you being here, is that you meet someone here, or you have a question you raised in the class so we can discuss it. So yeah, take advantage of it. Okay, so let's jump in. What is spatial transcriptomics? In one sentence, it's a new technology that measures transcripts or make transcripts seen. In biology, transcripts means RNA, not your grade, like we just talked about. So every cell has some transcripts, has some RNA. If we want to measure it in a way that preserves the spatial location of those RNAs in cells and in intact tissues. The jargon would be in situ sequencing, in situ technology. And we wanted to do it in high throughput, meaning we wanted to measure many cells and many genes simultaneously in the same tissue. If I just show you one picture, that's the essence of it. That's a slice of the mouse brain cutting in the in the sagittal section. Sagittal means front to back, cut it this way. Mouse brain, a bit different from human brain. And for all the cells that fall on that slices, I don't know, hundreds of thousands of cells on this slice, we measure 30 selected genes. Each gene is coded by, each gene is a type of mRNA that has been captured and represented by a different color. You see many different dots. Each dot is a gene and an mRNA, and you can see their location, where they are throughout the mouse brain. And that automatically reveals brain structures, which genes are expressed where, which brain region is which, and so on. Okay, you can think of it as a fancy staining technique, like staining people use to look at tissue, to reveal structure. This is like staining, but with molecular detail. We know which genes are where. Okay, and it's a beautiful image. So that's pretty much spatial transcriptomics. Any questions so far? So we're done with what is spatial transcriptomics. Why do we need it? Obviously, it looks like a very powerful tool just by looking at the pictures. But why do we need it? Let me dive into the basics just a little bit, maybe linger on it. And I wanted to encourage questions. Here are a few questions to begin with. Why do we need to? or why do we want to see so many mRNAs displayed in tissue sections like this in cells and tissues? Might sound basic to biologists if you're working in the field, but this class has also targeted those who are interested in biology and who study physical science or engineering. So let's just go all the way from the beginning. Why not measuring water and lipids? Again, water is the same everywhere. Lipids, there are a few different types, but our cellular membranes has it. It's not that specific in a sense. Why not DNA? DNA contains all the information you have passed on from one generation to the next. So it contains all the biological information you will need to develop a whole body. But it's the same in every cell. So if you measure DNA throughout the brain section I just showed you, it will be pretty much the same everywhere. There will be certain error here and there because of replication in the development, which may have some implication, but by and large, it's the same. Why not measuring proteins? Proteins are different here and there, and they carry a lot of different molecular functions, biological functions. But it's hard to measure it comprehensively. You can develop antibody to target one type of protein or a few types of proteins, but not comprehensively in one huge group to study thousands and thousands of proteins in the same tissue. So we are still not there yet. So remember, what is COVID vaccine? It's not protein-based. There are protein-based, but the most successful ones are based on mRNA. And the reason is that people know how to make mRNA and design it easily, much easier than proteins. Why in cells and tissue versus in the biochemical reactions happens in the tube? But we do want to preserve the tissue or the spatial location of those RNA cells, because I guess if you imagine we go back to the sections, ring sections, if you don't preserve the spatial location of these structures, all different 30 selected genes will be mixed up in a tube where you can measure, you can still count how many genes are from, how many copies are there for each gene, but you won't review the spatial organization. And that's key for biology, right? And the last question, why high throughput? Why do we need to measure so many genes? Why do we even care? We can study one gene at a time, and there has been technology to do so, and we can study one cell type or cell line at a time. But we really, in order to understand many different biological questions, we need to measure many genes in many cells at the same place, because that's how biology works, how multicellular organisms work. If we just consider how many genes are there, how many genes are there in the genome? Anyone who study bioinformatics?
20 to 30,000.
Yes, 20, 30,000 protein coding gene, faculty member. Okay, and then how many cells are there in the brain? It's hundreds of millions of cells in the human brain. Anyway, I'll show you a picture about that later. So because there are so many cells and genes, they coordinate their actions at the same time in the tissue. You really need to have a way of seeing them in action in order to understand them. That's important, right? If you break it down one gene at a time, a different gene for a different sample, you lose all these possibilities to study. So that's why people think this is a powerful way, and indeed, these single-cell transcriptomics or spatial omics or different omics has transformed biology in the last decade. One review here by Steve Quigg has shared his thoughts. Essentially, what we are doing here is to treat cell as a bag of RNA. A cell has lots of RNAs contained within it. If we can measure, we can just count how many RNAs how many copies of this RNA and that RNA are in each of those cells, we can understand a hell lot about cellular biology and all types. So essentially, we just wanted to generate a table, right? Rows are different types of RNA, cells are cell A, B, C, are you can think of different cell each as a person have took a different bag of RNAs from a selected population. So this is enabled by the reason. So a decade ago, there's no way you can try to generate a table like this. But over the last decade, technology has developed so rapidly. Now it's routine to generate this type of data table. That's enabled by single-cell genomics. We will talk about the techniques here, spatial transcriptomics in particular today. And that in turn transformed biology into a quantitative and computational era. So now given that it's a table, you can apply all different statistical measures, all different computational tools at your disposal to try to figure them out, understand better about it. Okay, so to give you an example, people have always wanted to study cells for centuries, and people discovered cells over a century ago. And in the nervous system, it's a bit late. It's just about a century ago, by Cajal. He's the founding father of modern neuroscience. Back then, they used advanced technology, staining technology called Golgi staining, which can randomly stain brain cells to review their shapes and sizes. The trick is that you can't stain all of the cells all at once. If you do that, it will be a blurry image. You can't see anything because the brain's tissues are too dense. But if you just, for some reason, randomly stain some cells, you review their full shapes and sizes. And it looks like each cell is different, has different morphology, and that opens up modern neuroscience and the way we try to understand cells. So over time, people see cells as different things. Initially, we see through their morphology. It looks like a tree, or they must be engaged in some connections with each other. Later on, we know they have physiology. They transmit electric signals, and we have ways of measuring electric signal for different types of neurons, and they have different types of signal being sent over there. And later on, we can categorize their cell location. Different cells are located in different places in the brain. They talk to different other cells in different parts of the brain based on anatomy, connectivity. In the end, molecular biology came along and showed that different cells really have different molecules. By molecules, in part, we mean mRNA molecules, the focus of today. And molecular signature underlies all aspects, pretty much all other aspects of cellular properties. The reason that cells develop into different shapes, have different electronic properties, talk to some other cells in different sets of cells is because they have different molecules, or in part because they have different molecules that each is responsible for different functions. So if there's technology that allows you to survey all these different cells, different molecules they express, that will be powerful, okay, for neuroscience. But not only for neuroscience, it's a general question in biology that how do cells grow into different types? We just saw a few different types in the nervous system, but there's all different types of cells in different tissues in our body. And how do those different types organize themselves and then fulfill some coordinated biological functions, right? That's such a complex question to be asked, and it's hardly imaginable how we can even begin to study it. And there's different incarnations of that general question. For example, in neuroscience, we study how does the brain, which consists of neural circuits, give rise to the mind? Why do we think that way? In development, we study how the cells divide, differentiate, and organize to form complex tissue, like the brain tissues. In cancer, for practical reasons, we also wonder what are the biomarkers of those tumor cells? How are they different from normal cells? Are there different types of tumor cells and needs? So we need to develop different drug targets for each of those. Okay, so what's the key to answer all of these? I guess for now, we need to take different steps to answer that. But it's been clear that it's not enough to understand just the molecules. Because we talked about molecules, different molecules are in different cells. But molecule is really just parts. We need to understand how those molecules organize into organisms. The organization part is key. Why do we think it's a key? Just to provide one example, if you think molecule is so important or by themselves, you just count how many genes are there in different species. In this post-genomics era, we can sequence a lot of different species. It's not a big problem. So for example, E. coli bacteria cells have 4,000 genes, right? And then chicken has 17,000 genes. Oh, you may think, oh, 17,000 is a lot more than 4,000. But really not much if you compare the difference between a chicken and a bacteria. That's orders of magnitudes of complexity difference. And if you think chicken is not enough as a comparison, a convincing comparison, we human just have a bit more genes than chicken, right? If we are satisfied, well, we have 20,000. Actually, water flea has more genes than we do. Rice have more genes than we do. Okay, so the reason that we develop such complexity is not just because we have a bit more genes than bacteria or so on. Okay, it's because how they organize, how different gene, different sets of molecules happen to be expressed at the right place in the right time, at the right time at the right place, so that somehow it all works out. So in a sense, biology exciting, its design problem cannot be broken down into similar parts. All living organisms are alike, created from the same parts. By that I mean, despite the vast difference between E. coli and humans, we all use the same machinery, molecular machinery. The DNA sequences use the same letters, A, T, and G, and Cs. We use the same 20 amino acids, and the basic machineries are similar, and certain genes are remarkably conserved between lower species to the next. But it happens that these complexities are all because of some innovative organization of those basic molecules. Okay, so it's very different from how we generate, say, make a car, you need different parts. You make a plane, you need a different set of parts. If you make a table, it's yet a different set. Versus in biology, muscle cells, it's the same as neural cells in a sense, all the genes are the same. You just need to count how many copies are present in each one. Right? That makes our life easier, but this puzzle hopefully makes this puzzle more interesting to you. To give you another example in neuroscience, there's this problem called the genomic bottleneck. If you just consider, also because this is a quantitative class, this is our first quantitative analysis or exercise. We can do it together. Okay, from a bit of information theory as an introduction. So let's consider what's the size of the human genome? How many base pairs are there in the genome? We now have sequenced thousands of human genomes. Everyone are pretty much the same. How many are there? How many base pairs? Anyone wanted to take a guess? Or you can ask ChatGPT, Perplexity. But so there are about 3 billion base pairs. So each base pair is one of the options A, T, or G, or C, and it goes along one dimensional on and on for 3 billion base pairs. And how much information are there in the human genome? By information, So that's where information theory comes in. Okay? How do we count? How do we store information? So we usually store information in bits. One bit is just either 0 or 1. It's an answer to a yes or no question. Okay? So if it's no, you say 1. If it's yes, you say, yes or no, 0 or 1. So one bit can represent one yes or no question. So how many bits are there are needed to store 3 billion base pairs of human genome? It turns out to be an easy question because just think about one base pair at a time, right? For each base pair, you have 4 different choices. DNA has A, T, G, or C. You ask, is it A, is it T, is it G, is it C? How many questions do you need to ask? How many yes or no questions do you need to ask in order to locate one of them, to figure out, are you one of the 4 for just one base pair? You need 2. You may think you need 4 yes and no question. Yes, you can say that counts 4 bits. But that's not the most efficient way of doing it. The most efficient way is, are you on the left side or the right side? Okay. If you're on the left side, are you then further on this or this or this or that? Anyway, so you need, rather than 4 bits, you need 2 bits of information to store one base pair. So for example, in computer code, you can, for example, represent A as 0, 1, T as 0, 0, G as 1, 0, C as 1, 1. Okay, so 2 bits can represent one of the 4 letters. That's 2 bits of information. So how many bits in total do we need to represent the full genome? It's just 3 billion times 2 bits, 6 billion bits of information there are in the genome. Okay. How many neurons do we have? I just talked about hundreds of thousands. And then how many, hundreds of millions. How many information is needed to specify the wiring? Okay. Let me go up to this part first. Hundreds of billions of neurons in the human brain. That's more than the number of base pairs in the DNA, right? Every cell has the same DNA, only 3 billion base pair, but the number of neurons is much more, number of cells, much more than that. And if we consider this, the last question, how much information is needed to specify the wiring of neuron? What do we mean by wiring of neuron? Neurons are sending out these processes. They have antennas, we call dendrites, to receive information from other neurons. They also have axons to send information out to other neurons. So the connection is not random, because if you're born to function, to know how to interact with the world. If the neurons are just random, it's not going to work. But there's some molecular code that allows the neurons to know how to connect to other neurons in a stereotypical way. That's the wiring problem. But how much information do we even need to? So if you want to design a blueprint of neuronal connection, like electrical circuits, how much information do we need to store that information? Right, that's a question. Any guesses? Or how will we even begin to do that? We need a big table. Again, that's information can be stored in tables. So, for example, we need a table where every row is a neuron. So for neuron I in this row, I represents one of the neuron, 1 out of your 100 billion, right? Every column is also a neuron, the same set of neurons. So I will be here, I same neuron, and or we could look at a different neuron J, right? So you might say that if neuron I is connected to neuron J in some way, their processes meet, we record oh, here, one, or whatever, they are connected. Otherwise, if I and j are not connected, we can store a lot of zeros there. Say they're not there, they're a different part of the brain there. They don't talk to each other directly. So this table can be filled with lots of connections, right? But this is how you can store an activity matrix. But how large is this matrix? Just simply count. You have the number of neurons is hundreds of billions. So you have hundreds of billions of rows and hundreds of billions of columns, right? That's hundreds of billion times hundreds of billions. You could try to store it more efficiently if you make some assumption saying, oh, certain neurons are not going to talk to many other neurons, or the structure is sparse and you have better way of doing it. But really you need way more than hundreds of billions of bits for sure to start with, okay? So what's the bottom line? So the genome is 6 billion bits. The number of information needed to specify this is much, much more than that, okay? 6 billion bits is surprisingly small. It's less than a gigabyte if you're familiar with computer storage. In your phone, you can store perhaps 200 gigabytes. So you can store 200 genomes in your phone. You can easily store 1,000 genome in your laptop and do some analysis, right? But there's no way you can store even one set of wiring diagram precisely like the way we show it here in your laptop. But how did it even work? The genome is responsible to specify the wiring program after all, right? And then also they're responsible to develop other parts of our body. The bottom line is the genome is strikingly small for what it does. And why is it so small? What does it imply, right? So are there other examples like this? It turns out mathematicians have long figured out, or different areas of work. I happen to be familiar with math for a little bit. If you think about information storage, the best storage is when you understand a pattern. If you understand the pattern, you can store information much more efficiently than you apparently can. So, for example, here's a list of very complex numbers, right? How much information do we need to store that number? It's hard to calculate, but you need a full page. You need to write them down and they look random, right? But if you know how this pattern is being generated, it's actually generated by a simple rule. The rule says the first two number will be 1 and 1. So it starts from 1 and 1 on the top left. Then to generate the next number, it just adds up the two previous ones. Okay, so the third one is 2 because 1 plus 1 equals 2. And the one after that is 3 because 1 plus 2 is 3. And then you go on and on and on. Simply adding up the two previous one, you generate a more complex pattern. You generate yet a more complex pattern, right? So this is a toy model, or just what's the point? Point is complex pattern can be generated by compact rules. So if you know how the rules are generated, you can just write that rule down, and it automatically stores all the information to review that pattern, right? Biology must have done things this way. Why is that? Because there's not enough space in the DNA to specify all these patterns, or that wiring diagram, how the heart is being built, a blueprint. Not that way. But it must have some compact rules to allow them to generate these very complex patterns, right? So complex biological organization have to be generated by much simpler developmental rules given its limited space. But many of these rules are unknown so far. We know a few places here and there what are the rules to generate this pattern, but largely speaking, it's not understood. Why is not understood? We don't understand the pattern first. But now we have a technology to let us directly looking into the patterns, and hopefully from that we can then derive the rules that must be there. So that's what I think biology at this point is very interesting to biologists or to physicists, to those who study engineering. There's a rich ground of question you can explore, and the tools we have are about ready to ask those questions. So let's return to this cell as a bag of RNA. And we wanted to figure out which bag of RNA each cell has. How can we, then the next question will be, how can we measure RNAs in a cell? How many types of RNAs are there? How many copies of each type of RNA are there in a cell? That's the task for today. So we figured, we talked about what is spatial transcriptomics. Why do we need it? And hopefully that's the case for why do we need that. It's an exciting technology we can use to answer many questions about biology still unknown. The next we will be focusing on for the rest of the class, I guess, how can we do that? So the question reduces down to how can we develop a technology, or technology has been there. How would the technology being built to address these problems? Okay, then we will read two papers next. So I will give a bit of high level how, and we will take a break, and after that we will read papers in details. Okay, how can mRNA be measured, be seen? So you can, method one is by sequencing. So if you take a cell has a lot of RNAs floating inside. If you break the cell membrane, capture the RNAs, and then amplify it and sequence it, you can know for each of the sequence, each of these mRNA, what are their sequences, and then understand which genes they come from. That's one way, by sequencing. This way, you capture all different types of mRNAs or genes, but you do not retain spatial information. Okay. The combination of this technology is single-cell RNA sequencing that has been used nowadays very everywhere. So the way you do it, if you wanted to study a tissue, you dissect the tissue, you isolate the single cells, so all the spatial information will be gone, because you one cell at a time, and then capture them, release their mRNA, and then sequence. So you get single cell RNA sequencing. Okay, and after sequencing, you get a big table of for each cell how many genes they express and so on, then do analysis. A different way is by staining and imaging. So staining means if you have a cell, you can clear it, but you hold it in place in situ. But you introduce some probes that carries fluorophores to specifically binds to the gene you wanted to review, and then doing imaging or light microscope to let those genes or mRNA molecule emit a light signal. So you can then count, oh, here you have 3 different dots, 3 copies of mRNA in this cell. This method allows you to capture one type of mRNA, one gene at a time, and it preserves all the spatial information. Okay, so 2 different technology. One capture all different mRNA, no spatial information. Here is one that capture all the spatial information, but you can only do it one at a time, because in order to distinguish which gene is which, you need to design probe that specifically binds to a particular type of mRNA and bind it and let it emit a different signal. If you want to study a different gene, you need a different set of probes, right, that binds specifically to them. Okay, so you can stretch the limit of this imaging technique by doing multiple rounds of imaging, stripping, and re-imaging again. So if you have a cell, in the first round, you can introduce a probe that binds to one type of gene, and then you strip it and introduce a second set of probe that allow you to study a different set of genes. So that way you can do, with multiple rounds, you can study multiple set of genes. But it's limited. We have 20,000 genes in our genome. You can have no way to do 20,000 rounds of stripping and imaging, introducing different probes. Cells will degrade over time and so on. And it'll take a long time to do that. What if we have a technology that bridges the two? One is staining, the other is sequencing. What if we can make staining more high throughput, staining more genes simultaneously? What if we can make sequencing spatial resolved? Sequencing preserve no spatial information, but what if we can? That's spatial transcriptomics. Okay, so there are two major branches of spatial transcriptomics. They all originate from their respective existing technology. So one is sequencing based, evolved from single cell sequencing. How can we make them spatial? And the other is imaging based. It evolved from this imaging or staining approach. How can we make them more multiplex, having more genes? So there are two branches of technology being developed along those lines. They are both called spatial transcriptomics. They have their pros and cons, and they converge over time in the last few years to try to, but there's different trade-offs or pros and cons for different technology. If you just represent different technology on this two-dimensional plot, on the one hand, you have, I guess, single-cell RNA sequencing, being able to measure a lot of genes but no spatial information. On the other hand, you have single molecule FISH, SM FISH, which is a staining method that allows you to stain one gene at a time, preserving all the spatial resolution, but only one gene at a time, okay? So over time, over the last decade, people invented merFISH from single molecule FISH that jumps from measuring one gene to more than hundreds of genes, a couple hundred genes. That's merFISH. On the other hand, people invented SlideSeq and all different technologies that allows you to also preserve spatial information by sequencing. That's SlideSeq. Also a lot of different variations on that. So there are trade-offs about multiplexing how many genes, resolution, spatial resolution. Is it single cell resolution? Is it even more? And throughput, how many genes, how many cells, sensitivity, how well we can capture the genes and cells in the tissues. There are these trade-offs. Different technology have different strengths and weaknesses, but we will focus on the two. The two paper correspond to the two. How does it work? And these two represents two major technological branches of spatial transcriptome. So those in the middle are spatial transcriptome. Okay, so let's take a break. After the break, we will read about the first paper, and then we will read the second paper, and that's for the day. We're at 10. Let's take a 10-minute break. Okay. If you have questions about this class, feel free to let me know. Also, for those of you online, you can type your questions in the chat, or you can unmute yourself. Just ask a question. Okay. And if during the break, so after the break, we will talk about this first paper. So if you feel like just start reading the paper, just go ahead. We will read about this Chen et al. 2015 paper after the break. So, yeah.
Tomorrow at 10 to 11, I have to take a Zoom call.
First.
So I can be here from 9 to 10.
No,
and then
no
from
worries.
11
Yeah.
On Thursday.
Yeah.
So I will try to be here as much as I can, but it will be a little.
Sure.
I'm not taking it for the grade, but
Yeah, yeah.
yeah. But the coding especially, I'm really interested in. But you will probably provide
Yeah.
those.
Yeah, I will
templates.
provide.
Okay,
Yeah.
good.
Yeah, if you are in the first bit of it, I think it'll be great. Then,
Okay.
Yeah, then
Yeah, I'll come
Hopefully.
back after the Zoom call.
Sure,
tomorrow.
sure,
Okay,
sure.
good.
yeah, let me know if you have questions.
Okay,
Yeah.
okay.
Hi, how are you?
Good. How are you?
Good. I just wanted to ask, did you take attendance by any chance? I might have.
I haven't. I think Eloy will come and take attendance, but if he doesn't, we'll see. Or just do it. Okay.
So I think some spatial analysis with the RNA found some gene specifically supposed to A, but it's like from the literature, right? They're supposed to express in cell template A, but it may be our analysis, sometimes I find also in cell template. And the interesting thing is that, the real world, the real tissue, these two cells have never close. So I found, it's quite a, some like a contamination. I think this is very real in single cell, right? But in spatial, it happens, how to solve this kind of thing.
Yeah, right. You're saying you have two spatially localized cell types A and B.
Yeah.
You find cell type A also expresses a gene,
Yeah, right.
and you might think, are you sure it's a contamination or is that?
I'm not sure. Actually, like this kind of gene, right? They're specifically expressed in cell type A because based on the staining and the literature, everything, it's very specific. But it shouldn't, in like another cell type. But
By a lot or just a little bit or? Okay, just
10%.
10%.
Yeah.
It's hard to tell. Like if probably two. How can you distinguish two possibilities? Right. Possibility one is a genuine signal. People are just really less sensitive approach. Not previously you didn't capture it. Capture the predominant pattern, but that's one possibility, right? And then the other possibility is like it's some contamination of B cell transcript to A.
Yeah. So let me show you, maybe this is more clear, and then like in the spatially, they overlap together. Let's say, when you do the cell segmentation, maybe they recognize this one as one cell, but actually they have some cell body of another cell type.
Okay, you think it's a segmentation? I see. Segmentation area fits into this, but that do you have an estimate of how much segmentation area? Yeah, that's one thing. The other thing is, again, one thing is to try to reduce the level of noise as much as possible. Say, what if you improve segmentation, maybe manually a few cells? Would that resolve the issue at all? That's one thing, right? Or then over time you can. Oh, sorry. Okay. One thing. The other is, would you be able to do I don't know, complementary orthogonal experiments? Say, if you rather than spatial, because segmentation is a spatial process. What if you do some single cell? They will still have contamination, but it's a different contamination problem. I mean, that in their contamination is about all the cells you collected release some of there's ambient, they call it ambient mRNA. That is the same for all cells rather than having a spatial. But there's tools to correct for that too. If I don't know, I would imagine, but it depends on how much, if there are tests you can do too. But there's definitely tools. So noise will be presented in different ways by different technologies, but biological signal hopefully develops.
Okay. So one more comment in medium. So it's, I mean, besides the default cell segmentation, do you have any like training?
I don't know what's the default. Is it cell pose? Cell pose is what people are now using. Used to be this cell pose 2 or something like that. Right.
Okay.
And you can check it once you use it, it generates results, right? And then you check the segmentation results whether they make sense.
Oh,
Yeah.
thank you so much.
Okay, let's get started. So in this figure, it captures fields arranged by purity. On the right side are the fields that are more pure, and on the left side is less pure by some standards. I think it captures a certain mindset of reductionists. So sociologists look away because psychologists think sociology is just applied psychology, study many brain versus one brain, the many mind versus one mind. And then biologists think psychology is just applied biology, perhaps applied neuroscience, because mind derives from the brain. And then chemists think biology is just applied chemistry because, cells are just molecules. And then physicists think it's all physics because, all different chemicals are just electrons, atoms, and interacting with each other. The mathematician thinks you just, everything, the laws of nature are written in math, in math. But anyway, so that's one type of thinking. But there's a different set of thinking, I guess. Here's a quote, in studying life, you keep diving from higher levels to no lower ones until somewhere along the way, life fades out, leaving you empty-handed. This is by a biologist taken from a biography. Anyway, people grappled with this question. If you want to keep going back to the origin, to the basis of what's going on, do you at some point lose it already, right? If you study biology, at what point do you stop studying biology? In physics, people also grapple with this question too, called more is different. There's a debate in the 70s, and back then physics was entering, it was still its heydays, but there's some cracks in the field already, with some people claiming that our field is the true physics, the other are just applied physics or engineering. But here is one physicist saying more is different. His argument is adding more similar things together, interacting with each other, will emerge a new pattern that's different from the basis. So that's a different view from the reductionism mindset, saying everything is just biology, then biology just chemistry. Okay, so what does it have anything to do with this class? So before spatial transcriptomics or single cell omics, people usually study one gene at a time, one cell at a time. You often miss the part of a big picture. But here we really have technology that enable us to study a bigger organization that allows you to ask better questions about biology and how they work. Okay. So reading sessions, and here's the plan. I will give you 10 minutes reading this first paper just by yourself. I will 10 minutes is short. I will tell you where to look to read and which part to understand. And as you do that, please take notes on where you are confused about. What are any parts doesn't quite make sense to you. Okay, so then we'll have 5 minutes pair and share. In the beginning, you talk to your neighbors. You will talk to your neighbor again after reading the paper, just to have a short discussion and together come up with at least one question among your group, among your neighbor. And after that, we will collect all the questions from you, and we will try to address them, have a discussion. And in the end, we will have a mini quiz, again, not graded, just to test whether we understand the paper, okay? The point is really just to try to understand as much as possible, but note where you don't understand. Okay, that's very important. Okay, so for the first paper, Chen et al. 2015 Science, I have sent it to you yesterday. If you haven't got it, let me know. A PDF. It's the first PDF. That's the paper that introduced MERFISH in 2015, 10 years ago. Since then, a lot of MERFISH papers have been published after that. Very influential paper. So I want you to read just the first page of that paper, introduction, rationale, high-level summary, and try to understand figure 1, the first figure, in the main text, not on the first page. So you'll read the first page and try to understand as much as possible figure 1 in the main text. Okay. And here are some questions I consider. And as you do that, try to come up with questions. Okay. So I will give you 10 minutes to do that. And we will come back at 10:27. Okay. Okay, so that's 10 minutes. Now let's find your neighbor and just talk about what you get from it, what you didn't get from it, and for about 5 minutes, and then from each group, I will collect question, at least one question from each group. Okay, so maybe you three can go together and figure out what you don't understand. Okay, you guys you can self-organize like the two of you and the three of you in the back, you could go together. Come up with at least one question in the end. You might have more, so the more the better.
I didn't quite understand.
That means a lot of questions.
I'm like, I was just trying to put a couple words down. What it is. Thousands of mRNA species that single cells from. My understanding is I think these articles. Yeah, because this has. And then a couple of others. Yes. And to the end, whatever shires. I don't decide sometimes to figure out.
Yeah, and then second, I wanted to make like the word next digit. So it's that. Keeps going. So like figure out what it is that just and how how it's I was like that make it I guess I thought it was like a cluster off. If there's something that's like done like live, right?
Yeah, I don't.
Yeah.
They said that to read the first page, but then look at the figure. Or maybe I didn't even listen to this.
Oh, do you think this one was more like an audio level?
Yeah.
This one is very.
I tried, but I don't.
Yeah, I read this one too.
Yeah.
This because this is like the summary here. So, yeah, so I think what they're doing is. That basically kind of this what I was asked is like so for does it take two images to understand the first I think like it's the first base pair, right? Because like right, it's like that 2 bit.
Right.
So then I think say 1 euro is right.
I see,
I
okay.
think that's what they're doing, and so basically, just divides the data 2 bits, and so, so, and it also will give you, the length of the new sometimes, I think. I'm the length of your side, that gives you, and divided by 2.
I think this is a really good question. We could, well, because that's gonna be out. Without really reading the text, because I thought maybe the image per
Oh, yeah, so I think the process is just to understand that it's like the process is just as you think about it, right? So like.
But they have actually a 6 they have 6 months application,
right? So, which is why I'm also because if I'm the way I understand it, these are 2 rounds just quickly.
the profile is in here.
Yeah, I think yeah. It's let me go. But But they're not using blues. Yeah, she was talking about this. When I do in-situ, I do like, I use a probe that's specific to a gene.
Yeah.
It's long,
Yeah,
but I don't think that would use for a ton of
Yeah.
rounds.
Yeah.
And so I think it's just like these little short little bits, but I want to know how do you deal with these problems? So that fun.
Yeah, you get action.
Because there's also a chance that the probe is like.
Okay, I'll give you one more, maybe 30 seconds to finish up. Thanks, Patrick. Sorry.
I think I want to know, I think I want to know how long the probes are.
Yeah.
Right?
If
it's just, yeah, like how, because it would make more sense, you know, if that targeting. Would really be like. Nucleotides long or not. Yeah, that's really
Yeah.
But then how many? Like even if it's 2 hour, you speaking that out. Like later.
Yeah.
Yeah. Add stuff with it.
I guess we have more questions. I have more questions.
Yeah.
Okay, yeah, that's great. Okay, now let's collect the question from each group. Or maybe you guys can get started.
Okay,
Ah.
we have a question in figure 1E panel.
1E panel.
We don't apply, like, for example, the blue prob. You can identify from the 3' or 5'. And does that matter? Yeah,
Blue probes, blue probes.
yeah, but also in 3'. Yeah.
Okay, yeah, just make it, yeah, I'll take the blue probe as an example. Thank you.
Yeah.
Any other questions?
Between each image, are they adding probes?
Adding probes,
Like.
Is it the same prob or?
same or different?
Yeah, different.
Or maybe just adding the fluorescence?
I see.
Yeah.
Between imaging.
Yeah.
Good questions. Okay, both of these are about the implementation of this technology. Okay. Do you have anything to add or anything else? Okay. You guys.
I have a lot
Yeah.
of questions.
Sure.
No.
Great. That's great. Yeah.
But the ones I want to ask are more like tangible, like how long are these probes? And is then the length that you're looking at, the length of the RNA that you're looking at, is that just n divided by the length of the probe?
Length of RNA versus their relationship with the length of the probes and how long are the probes.
Yeah.
Okay, got it. Anything else?
So many other things, but we'll
Okay, I'll come back then. Yeah.
Well.
Oh, yeah, maybe you.
One, this
uh.
is maybe fundamental. I think that this paper assumes that you know what the Hamming code is. I have no idea what the Hamming code is.
Hamming code.
So I don't know what the modified Hamming code is.
Hamming code modified. Okay. Okay, that's more conceptual. This is about the implementation of it. Yeah.
Yeah, so I actually had a very similar question because I understood some of the, let's say, solutions that they found to identify incorrect for errors, but then it's not super clear to me how precisely that work. Like I understood that they keep the number of 1 bits low and that's okay, but then I don't understand how do they realize that there was a misreading and correct for that.
How to detect and correct correct for mistakes. Okay. And that's related to the code and stuff, right? And do you have anything to add? Okay. Okay, last group in the back. Any questions?
I guess I like figured it out while looking at this, but my question was how do they get rid of the probes without getting rid of the, how do they get rid of the fluorescent probes without getting rid of the mRNA that's attached to the DNA between every single round?
I see how to strip the fluorophore without removing the probes. Okay, anything else?
I just don't understand the error correcting flow.
Error correcting, okay, good. Any major things to add, or you want to hold it for the future?
So
Okay.
the binary words, I don't quite understand how they construct the binary words.
Binary words, yeah.
Yeah.
Okay, so roughly two sets of questions, I guess. One is more conceptual about the design, the other is about the implementation. Let's go from the design first and then move down to the implementation. Some of it I may not know for sure. Check my answer. Okay, so what are the key ideas about merfish? So first of all, merfish is a fish technology, fluorescent in situ hybridization. It evolves from what they call as SM fish, single molecule fish. Before merfish, how do we do probes and fish detection? We design a set of probe specifically for one gene at a time. So with one round of imaging, you get one gene. With n rounds of imaging, so rounds versus number of genes. Okay, so before, if you have n rounds of imaging, you can get information about n genes. The key idea here is what they call as combinatorial barcoding of genes. By that, if you do n rounds of imaging, you can get how many genes in principle? 2 to the n, 2 to the power of n of gene minus 1. We will talk about why it says minus 1. So for example, right, if you get 10 rounds, if you do 10 rounds of probes, before you can see, you can image 10 different sets of genes. That's already a lot, and you only get 10. But now with 10 rounds of code, in principle, you get 2 to the 10 minus 1 is can be ignored. 2 to the 10 is about 1000, 1024. You can get thousands of genes being labeled this way. So that's what's called combinatorial barcoding, or combinatorial labeling, right? Is this concept easy to understand? It's used in our everyday life. If you go to a supermarket, every item has a barcode. It's the same barcode people use everywhere, and that's about 10 digits, 10 different 10-digit long barcode that can be used for all the items you can come up with everywhere in the world. That's because it's combinatorial, right? So here you are limited in the sense that in imaging, you only have a signal or no signal. I guess you can try to measure the intensity, but it's quite messy. So you either have one or zero in one round of imaging you get. That's binary, right? In a certain point in space, whether this point lights up or is dim in this round. That's binary code. And then by combining multiple rounds of imaging together, you get a barcode. Is it on or off in the first round, on or off in the second round, and so on and so forth, for the same spot in space.
So the imaging is done once per each what?
Imaging is, let's take a look at the figure 1a probably, right? They have an illustration. So for this cell, they have a few different RNAs, right? Here, each line is an RNA. Let me blow it up. Okay, so they will image this cell 16 different times, but in the first time, they try to introduce a set of probe that specifically lights up these few genes. Right. So then while leaving out these other three RNAs, so then you move on to the second round. You remove the signal from first round. You remember it, but remove it. So the second round, in the second round, you introduce a different set of genes that lights up a different subsets of RNA.
So is the imaging done for each row set?
Yes,
Yeah,
yes,
right.
yes. Each probe set. Yeah, that's a good. Or in their words, each readout probe set. So you need to have something called encoding probe set in order to make this work. And then readout probe set is one readout probe per imaging round. So for the first set of imaging, you introduce one set of readout probes and image it. Then you remove the readout probes. You introduce a second round of readout probes and image it again, and on and on. Okay, so in each round, you're imaging all the genes all at once, but by just adding a bit of information to it, a bit literally in the information theoretical sense. Then with n rounds of imaging, you collected an n-bit barcode for each RNA you imaged there. That barcode is very powerful in distinguishing many different genes, because if you assign a specific barcode to one gene with 10 rounds, in theory, you can distinguish 1,000 different genes, right? That's a huge improvement from the previous way of doing it, right? If you were to design a system that can allow you to do this combinatorial labeling, its benefit is obvious, right? But how can you do that, right? That involves a lot of probe set. But one problem that immediately come into place is, so that's binary words, by the way. That's this A tries to show the binary words, how you can do that with imaging. But then that's only one part of it. Combinatorial labeling. It's simple, it's powerful, but simple. What prevents it? That only works in theory. In practice, it doesn't quite work. Why it doesn't work? Because here you have the nice thing of exponential growth, right? And to the power of n. With every new round of imaging, you get you can get two times more probe, two times more gene being sequenced. It's exponential. But what error also goes exponentially, if you think about it. Traditionally, say if imaging works 90% of the times, say you can detect 90% of your probes in one round, it's fine, you get 90%, 10% error. But what if you need to get n rounds of imaging and you are counting on all n rounds being correct in order to know which gene is which, right? That's a big problem. It's not enough to work 90% at each step. Then error also accumulates. How do error accumulate? If you get 90% in the first round, by the end of n rounds, error also accumulates the power of n. So 90% time 90% time 90% time 90%. By 10 rounds, the accuracy will be very small. You pretty much just get garbage, because in any step, the likelihood that you make a mistake in any one of those imaging rounds is quite large. Okay, that's what these figures try to show. So panel B shows as number of bits or number of imaging increases, the number of RNA you can capture goes exponentially. The y-axis is in log scale, so from 10 to the 0 is 1, 10 to the 9th is huge, it's billions. It goes exponentially. However, the mistakes also go, or accuracy also go down exponentially, and misidentification rate also go up exponentially. In theory, it doesn't work because if you say by the end of 16 rounds, most of it are misidentified. By the end of 16 rounds, the calling rate is 30%. Most of the signals are wrong, right? So how do we address that? And so the black curve is this naive way of doing combinatorial barcoding. So they need to correct for error without improving the existing technology of making one round more accurate. You need some error correction. That's the second point, right? The two main points for Murphy, combinatorial and error correction. How do you implement error correction so that this error do not accumulate exponentially, so that if it's under control, but we can still image as some genes or more genes than 10, something like that. That involves Hamming code and modified Hamming code. The idea is this: if you, out of 10 rounds of imaging, if you can tolerate one mistake and correct for it, then you can improve your success rate by a lot, right? How can this be done? So if you design a barcode that leaves out a lot of empty spaces, so some barcodes, you don't assign it to any genes. You leave those empty spaces on purpose so that if you happen to see a gene has that code, you know it's done by a mistake. And perhaps you can correct for that mistake by looking for the closest possible gene that mistake came from, right? So just to give you an example, and the Hamming code, what does it mean? Okay, let me get rid of this. Each gene has a barcode. Hamming code is a measure of distance or difference between two different barcodes. Okay, so here we have two barcodes. Gene 1 is has a barcode 1011. Okay, we just did 4 rounds of imaging. It's on here, off here, on here, on here. Okay, and gene 2 is 1111. Their Hamming distance, HD, will be 1. What does it mean? HD means you compare the two barcodes digit by digit, bit by bit. Are they the same? If they are the same, there's no distance between them, right? But on the second digit, they're different. Their distance is 1. On the third digit, same. So their overall distance between the two barcodes is 1. So what if gene 2 has a code like this, right? Then their Hamming distance would be 2. Okay, because they are differ by 2 bits. They should behave differently in 2 different rounds of imaging, right? Okay, so what did they say about Hamming distance codes? They said they wanted to have a design such that any 2 genes differ by at least 4, by Hamming distance of 4. What does it mean? It means if gene 1 is 1011, gene 2 has to be 0100. Now they're differ by 4. Okay, they're different here, different here, different here, different here. At least differ in 4 rounds of imaging. But then, so if you take more than 4 rounds of imaging, in their case, they took 16 rounds of imaging. So the rest of 12 rounds, they could be the same, but they need to differ by at least 4 in 4 rounds of imaging. Okay, so why do we need that, right? Because if you encode genes and space out the barcode this way, there's a lot of empty barcodes in between the genes. So that if you made a mistake in measuring gene 1 in one of the rounds, say in round 4, for some reason, it didn't detect it. Instead of 1, you saw it as 0. Then you know, by the end of it, you collected barcode, you know it's a mistake because you didn't design a gene to have this barcode in the first place, and you figured there's another gene whose barcode only differ by Hamming distance of 1. We know that first you detected this error, then you can correct that error based on the assumption that it's probably you made a mistake in detecting this particular gene rather than it belongs to a different gene, because any other gene has a distance much larger than Hamming distance 1. Okay? Does that make sense? So yeah, so in order to misrepresent, for example, one gene with another, you need to make at least 4 mistakes, pretty much, right? So yeah, if you misclassify 2 with 1, you need to make 4 continuous mistakes in order to misclassify it. That is quite unlikely. So by that approach, you can reduce error, but at a cost of some space being assigned empty, quite a lot of space being assigned empty. But still, you have a large room to work with in the beginning, even if you assign a lot of empty space for the purpose of being error correction, you still get a lot. So rather than getting 1,000, you can get, what they get is 140 genes. They use 16 rounds to do 140 genes, which is less than the ideal of combinatorial labeling power, but it allows them to control the error rate at a level that is acceptable. So they simulated it. So what if they use different coding error correction strategies, right? And then what they ended up using is this pink curve, which uses a lot of empty space to get but still they can image hundreds of genes, but the error is acceptable. Misclassification rate is very low because you require this set of space up pattern. Okay, so Hamming distance, modified Hamming distance, error correction, code detection for mistakes. Any other question on the design part? Okay, that's pretty much it for the conceptual part. How do we implement this in the for probe design? Yes, that's the second part of the question. So that's the part for figure 1E. Okay. So there's one question. So let's read through figure 1E maybe together. That'll hopefully resolve some of these questions. So they show you begin with a set of RNA you wanted to detect from 1 to M, right? So first you need to introduce this encoding hive or encoding probes. According to this illustration, the encoding probe has this shape. And, so this is your mRNA. This is your encoding probe. What does it mean? It means you have one part of the encoding probe. It's the middle section, specifically designed to bind to the RNA sequence. Right? It has to be specific. So for this part, it's non-negotiable. It depends on the sequence of mRNA. You need to design a part that is complementary to that. So, and there's also 2 other parts. That's your choice. You can design whatever sequence that's not constrained by the biological endogenous sequence. Okay. So someone asked the question, how long the probe needs to be? What do you think? How long the probe needs to be in order for it to bind? Maybe how long this section needs to be in order to bind to a specific set of mRNA? What do you think? Is it two nucleotide long? Let me give you a few up. 10 nucleotide long. 100 nucleotide long. I mean, just order of magnitude. What do you think? Who think it's 2? Who think it's 10? Who think it's 100? It's between 10 and 100. Actually, it's 30. It should be 30 usually. Why is that? Because obviously 2 is not specific enough. There are many different 2 nuclei. How can you bind to a specific RNA, right? But how long is specific enough? You can do a calculation. The genome is about 3 billion base pairs. You need it to be long enough so that it's specific to a particular location in the genome. Usually that's about 17 nucleotide long. Any 17 nucleotide, usually you will only find it once in the genome. Okay, but in order to make sure, you need to be a bit more than 17, right? And your 20 will be in practice they used 30. I don't know in this paper, but in the subsequent paper they use 30. Usually a 30 nucleotide, unless its pattern is weird, it's repetitive, you only have one place in the genome to bind it. So that ensures specificity. Okay. So that's the encoding part. What's the length of the readout probe? It's usually 20 nucleotide long. Okay. Again, 17 is the bare minimum to make sure you are specific. You need to go above that. So they use the 20, and this is maybe 30 will be not, it will be good too, but I don't know. I didn't design. So and they have two parts, 20 on the left, 20 on the right. Okay, that's the encoding probe. Okay, so with the encoding probe, you achieved combinatorial barcoding. Why is that? Because this part of the sequence, you can't do anything about it. You just need to design a probe to bind to the specific protein. But this part that comes with it will not bind to the endogenous RNA. It will be reserved for future imaging. And you can choose whatever sequence you want in order to achieve this combinatorial design, right? You can design probe such that this readout probe will not be bind in the first round, or it will be bind in the second round, something like that. Okay, so you introduce many of those probes. Okay, so once these are set, so this encoding probes are done once and for all. You flow it to the tissue, it binds everywhere. Then you go multiple rounds of reading out. So for each round, it will be a different set of readout probe binding to different sets of these space you created. Okay, so to answer your question, again, we address how long are the probes, the length of the RNAs. Length of the RNA, for example, this one single binding site is 30 nucleotide. They said they usually bind 200 of them. So you design 200 different 30 nucleotide space that covers it. Usually, typical mRNA has the space needed to work on and for you to work on it. For some really short mRNA, though, there's a problem. Yeah. So, the mRNA usually is thousands of nucleotides or more. Okay. So and then this, is it the same or different probes between imaging? It depends. The encoding probe is done once and for all. It's the same for all rounds of imaging. Then in the subsequent imaging, you will introduce a second set of probes carrying fluorophores. That will be bind to either this arm or this arm, not the endogenous RNA anymore. And that probe is introduced differently. A different sets will come in different time, and each round will be different. Hopefully that addresses it. And in 1E, and blue probes can bind to both 3' end or 5' end. So a probe can either bind here or there. Does it make a difference? It doesn't. And they just wanted to have 2 spaces for you to bind, and that gave you more space to work on, right? Yeah, but it's your choice whether, say, on this arm you can design a sequence allows you to bind to readout probe 1. So it will light up in the first round. On this arm, you can bind to readout probe 2. So it'll light up in the second round. That makes sense? It's your choice. But basically, they wanted to show that after the introduction of the encoding probes, you just have a second set of probe. Now, readout probe that happen to bind to these places, and then you remove them. Then a second set of readout probe bind to a different place. A third set of probes bind to yet another different place, and so on and so forth. Okay. In the end, you just collected this big table, keeping track of for each mRNA molecules whether they are on or off in each round. Okay, that's pretty much it. So someone asked, how do you strip fluorophores away without removing probes? Yes, that's how they did it. I actually don't know how they did it. So it's some chemical link they designed at the junction between the readout probe and the fluorophore. So when you add it, you can cut the fluorophore and wash them out. So after each round, this will be cut, fluorophore will be washed away. The probe still stay binded, but that doesn't affect collecting signal for the next round. The site has been binded, but in the next round, a different set of probe will bind somewhere else too. Okay, hopefully that looks like it addressed all the questions here. Any other questions? Okay, so let's oh, actually there's one from online. How does one determine how many rounds of readout are optimal for detecting the abundance of RNA? How many rounds are optimal? There's a trade-off, it looks like, right? With more rounds, you can see you can image more RNA, more types of RNA, but the error rate will also be higher, and tissue over time will degrade. So it's really just you wanted to push as many rounds as possible without breaking things apart. In practice, they did 16 rounds, but there's no limit on how many. Nowadays, they can do 40 rounds, I heard then, but so anyway, so that's it. So okay, so
Actually,
yes, yeah.
so once a probe binds to like one arm, another probe can't bind again like that.
Right,
Okay.
can't bind it. For one thing, that's by design. A different set of probes are specifically designed to bind to a different place, right? Or you mean the probe with same sequence, they won't bind. Yeah, if one binds, there won't be room for another. Yeah.
So why didn't they elongate? Right, like if two is better than one, right?
Right.
If 2 arms are better than one,
Right,
you can just have
yeah.
40 on both sides.
Why don't they do this, right? Then you can bind more. Yes, there's design for that, and that's called amplification, signal amplification. One strategy is by that. People also go more crazy. Like you could imagine having one encoding probe like this, and then you encode on top of encoding probe like this. You encode on top of yeah. Some people invented rolling circle amplification. They just keep generating copies. Yeah. So a lot of different techniques. It's useful in the sense that it improves signal, it improves brightness, and there are a lot of literature on this on how to. This is the first version
Right.
from yeah. Things will get complicated really quickly. But you can see it's a powerful and very flexible design. But there's trade-off, I guess. If you make it too long, you won't get into the tissue, and yeah. So okay, let's move on to finish this up. So I would like you to go to this website, and if you if you can take out your phone, there's a mini quiz or mini poll, a few questions about this topic. If you go to kahoot.it and enter the game pin over here. See. Okay, so if you go to kahoot.it, enter the game pin 456485, and let's join a short quiz for what we just talked about. Okay. Okay, let's get started. You can still join in your mind, I guess. Okay, what does Murphish stand for?
Oh.
Yeah, just select.
Oh, I see.
Select.
I was like, I was.
Yeah.
Okay, I agree.
Okay, great. Oh, actually, yes. Yeah. Anyway, this is a control question, just to test it in uses. Murphish is the extension of which technology? That's right, single molecule fish. The next paper will be about single cell RNA-seq of this. What's the spatial resolution of Murphish? Now we are a little split. What's the spatial resolution? The right answer is single molecule resolution with less than micron in length. Because I guess many things can be. One is it came from single molecule fish. Right? Single molecule fish stands for you can measure a small molecule quite accurately using fish. So naturally, there's no problem having single molecule resolution in Murphish. It just multiplexes. Also, if you think about it, why single cell resolution? Although it's nice, it's usable for biologists for many purposes. Why it doesn't work for Murphish if it's single cell resolution? So if you think about it, a cell is a bag of RNA. A cell has lots of different RNAs, right? If it's single cell resolution, how do you figure out which dot is which? then all these barcoding scheme wouldn't be possible at all without single molecule resolution. That's key. So say if in a cell you have two molecules, one here, one here, you really need to know at every imaging run, this one is this, but not to be misunderstood as the other one. Otherwise, the barcodes will just mess up, right? So spatial resolution down to the single molecule stacked across many rounds of imaging is key, is a foundation for this technology. Also, it makes everything needs to be super accurate in order to make it work. How many genes can Murfish measure? 1 to 10, 10 to 100, 100 to 1,000. Yeah, 100 to 1,000. In the paper, they showed 140 and then 1,000 gene. Nowadays, it can really do a few thousand genes. There's no limit, no theoretical limit for that. Would enable it to measure many more genes than the number of imaging runs. Yeah, both are needed. Combinatorial barcoding and error robust encoding, both are needed. Although, yeah, I just chose one. Are two key things Murfish introduced to the field. What allowed it to avoid error accumulation? Error robust encoding, of course, Hamming distance. What do we expect to see if a gene was assigned the following barcode? 1 means on, 0 means off. Yeah, light up 4 times because there are 4 ones in their barcode. Actually, in their design, they call it modified Hamming distance 4, meaning every gene needs to light up exactly 4 times, spread across different rounds. In the end, last one is the following scheme allowed by Murfish: gene 1, gene 2, 2 different barcodes. We have 2 different barcodes, one for a different gene. Are they allowed to coexist in Murfish design? Yeah, it's not allowed. Why is it not allowed? Because this is gene 1, this is gene 2. Gene 1, the difference is only in the first 4 digits. The rest are just 0. So gene 1 is 1111, gene 2 is 1110. What's their Hamming distance? 1, right? That's not allowed because if you make just one mistake, you will mistaken gene 2 with gene 1 and so on. That's not error correction at all, right? So that's not allowed. Okay, so hopefully, okay, congratulations, MA. GG. Oh, great. Towering. Congratulations. Good job, everyone. So yeah, that's it for this paper. So let's take, say, a 5-minute break, then read the second paper for 5 minutes, and 5-minute discussion. So the next 15 minutes is at your disposal. You can do whatever then, but make sure to read a bit about the second paper, only figure 1. And at the end, we will just do another quiz like this without discussion. It's relatively easier to understand the second paper, the concept, after we went through this. It's a different technology, but more straightforward. Okay, so actually, let's come back at 11:30. We will do a quiz, and then I will let you go. Okay, and you can take a rest and do whatever. The second paper is this one. Sorry. Flight seek, and you only wanted to read the first part and understand panel A and B, figure 1 A and B, that talks about how they do it. Then
Set it up.
Okay, we will jump to the quiz at 11:30. So and if there's question from there, we can come back.
Either. I've been dipping animal models for you guys because we get scan the QR code a little bit better.
Oh, that's good.
There we go. Let's see, go ahead. Let's see how long.
See.
What black models since.
Okay, let's get started right away without any explanation, just to see. Also, yeah, let's just see how well we get. What does the slide mean in slide seek? Yeah, everyone gets right. That's great. You may not even need to read the paper. But that summarized the key of this design, which is quite simple. You tweak from single cell RNA-seq. If you're familiar with it, single cell RNA-seq is done by flowing dissociated cell into one channel, a fluidics device, flowing barcoded beads from another, with the hope that each bead will capture one cell. And those beads are pre-barcoded by a different RNA, DNA barcode. So those barcodes will capture all these stuffs inside the cell. So all the RNAs of the cell will carry also a unique barcode from the bead. Each cell is from a bit different bead, so each cell has a different code. So then after that, you can pull everything together by sequencing. The sequencing will tell you, for one thing, the material from the cell, which gene they came from. For another, what's the barcode that can be used to separate cells from each other. So slightly take that idea, but rather than flowing dissociated cells and beads into each other, you just pre-manufacture packed some beads everywhere on the section on the slide, on the cover slip. Then so at each spot, at each location on the slide, there is a different set of molecular barcode on the bead. And then you just transfer RNA from tissue right onto the section so that different parts of the tissue will be coded differently. Then you read everything else by sequencing. That's their magic. Okay. So that has also been a very powerful technique. Since slide C, there are many other different variations along these lines. It's the extension of which technology? Yeah, single-cell RNA-seq we just covered. What's the spatial resolution of slide C doing this way? Single cell resolution. Actually, not single cell resolution. They try to claim single cell resolution, but let's see what's realistic here. If you design it this way, what limits spatial resolution? Is it single molecule resolution that you can get? Obviously not. But you're limited for one thing by the size of the bead, so and how close you space between them on the slide, right? And in the paper, let's go back actually. Let's go back to the paper. On panel 1, they show you the design. They pack these beads onto a slide, and the size is 10 micron. So the resolution can be no higher than 10 microns, I guess that's one limit. But what are other limits? For example, RNA diffusion. When you transfer the tissue onto a slide, will those RNA gets mixed up a little bit? How much they will diffuse? Those kind of things. So you can get resolution up to 10 micron, which is one cell, which is more than one cell. I guess about one cell, but some cells are larger, some cells are lower, but and plus diffusion and everything else, you get the resolution of a few cells rather than one cell. So every spot is a mixture of maybe one cell or a few cells. Okay. That's a key difference between this and merfish. Okay. How many genes can they measure? More than 10,000 because there's no limit. It's a genome-wide sequencing technology. Any molecule that's there, you can sequence it. Because we have more than 10,000 genes typically being expressed in a cell, if done well, then you can capture all of them. There's no limit. What allowed SlideSeq to retain spatial information using sequencing? Yeah, that's clear. Using pre-barcoded beads, what limits the spatial resolution? Yeah, bead size. Actually, nowadays, people use more and more tiny beads. That there's recently. So initially there's Visium. Now have Visium HD. There sort of fewer lengths. And stereo-seq and all others. All about the size of the beads. What can affect data quality? Yeah, it's efficiency. If you don't capture mRNA in the first place, you don't get it. Efficiency is a big limiting factor for this to work. On the other hand, it doesn't worry about tissue autofluorescence because you resolve the tissues afterwards. But for MERFISH, it's the other way around. MERFISH doesn't have to transfer mRNA out of the tissue. That's the plus. On the other hand, because you're working within the tissue, you need to deal with all different artifacts in tissue, autofluorescence and others. That's MERFISH. So you can see the trade-off between two different technologies. So that's it for the day. If you have questions, let me know. I'm gonna stay here for a while. And I also have a third paper. If you're interested, you can read it. That paper kind of blends together the two distinct technology together a little bit. So we talked about sequencing versus imaging, but people eventually also combine them, use it together. So there's also different variations along those lines. Okay, thanks for those of you online. I will see you tomorrow, and tomorrow